To another Bio 100. We're going to jump in today and cover a few new topics. So last time we introduced you to genetics, Mendelian genetics specifically, and we're going to kind of carry on with that a little bit today. But what's cool about today is we get to kind of tie our last few subjects together. So we're going to kind of tie together this idea of meiosis, genetics, and then add a layer on it today. And this is kind of a good time. At the very end, we'll go through and we'll watch a little YouTube video, which is a good opportunity to review what meiosis is, and kind of hopefully that'll help jog your memory, and also at the same time help you see what we've been kind of leading up to and how meiosis fits into genetics. So with that, I think I'm going to close myself out here. We don't need me for more of this. We're going to go ahead and get started. So last time I mentioned to you that we were going to talk about Mars and Marsa. So I used to do a genetics thing here at BYU where I had students produce their own gametes, and then students walk around the room and cross their gametes with others. And as you can imagine, at a place like BYU, that didn't always go over so great. I actually had some married students who were super uncomfortable with it. So I came up with this. So this is Mars and Marsa, and these are two aliens that are obviously found on Mars. They were discovered there, and turns out that their genetics were just like human genetics, and so this is what we're going to do. So what I would like you to do is take a look at this, okay? And you're going to have to draw this out. So get out a piece of paper. Go ahead and hit the pause button. I'll give you a few seconds, and pull out a piece of paper. Okay, so welcome back. So here's what you want to do. On that piece of paper, you're going to want to draw out essentially here. So I mentioned that Mars and Marsa's genetics work just like human genetics, and they do, except for they only have 4 total chromosomes. So they have 3 autosomes, and they have 1 pair of sex chromosomes. Okay, so a lot fewer chromosomes than we have. And so what you're going to do is you're going to go through and you're going to assign Mars and Marsa their genotypes for each one of these genes. Okay, so for example, chromosome 1 has 2 genes on it. It has the sports watching gene and the athletic gene. And so you're going to code them here. So if they are dominant, homozygous dominant for sports watching, they love them. They'll watch any single sport out there. If they are heterozygous, they're just all right. If they are homozygous negative, they can't, homozygous recessive, they can't stand to watch the game. Okay, so you're going to decide what that is, and then you're going to come over here. So let's say that you decide, and this is Mars, right? Because Mars has sperm, so here's the sperm. So you're going to come in here, and you're going to decide that Mars thinks that sports are just all right. Happy to watch them, doesn't care if he watches them or not. So you would mark big A and little a on his chromosomes. Okay, so you're going to come down here to athletic, and you're going to decide that Mars is also mildly athletic. He's okay. So it'll be big B, little b on that one. And then chromosome 2, you get to decide whether Mars is artistic or musical. Okay, so it turns out Mars is very creative. He can play an instrument pretty well. I don't know what instruments they're playing on Mars, but Mars can play them. Okay, and then you've done the same for chromosome 3, and then down here for chromosome, for the sex chromosome, now Mars only has 1 X chromosome, right? And so if it's a guy, he can only carry 1 gene, and that gene that you've decided here is that Mars is fairly calm and even. Now, you can vary from what I've coded here, right? This is just how I coded it. And then, of course, what you're going to do is you're going to take these chromosomes and you're going to walk them through mitosis. And we all remember, what's the end result of mitosis, right? It's gametes, and those gametes have a single copy, right, of each one of the chromosomes here. And so, if you run them through meiosis, you would have AB here. You would get this chromosome that came through. You get this chromosome that popped in. Now, actually, there's only one option in terms of which chromosome can come over here, right? Because he was homozygous recessive and homozygous dominant. And then here, this sperm right here is actually going to have the female chromosome. So X is going to come in here with its associated locus. Okay, and then you can make the rest. So there's a male chromosome here, another male chromosome, another female female carrying sperm there. All right, so now you've got these. Now hold on to it, okay? Hopefully you've drawn it out. You've coded it up exactly the way you want to. I'm going to give you a second. Go ahead and hit the pause button if you haven't done it on your own yet. It's really important that you do because you're going to need it in a second. So go ahead and hit the pause button and get that done. Okay, hopefully you're back. Now we're going to do the exact same thing with Marsa. So Marsa is female, right? So she's going to have 2 X chromosomes here. So you get to decide, you know, what she is and how everything works there. So you've decided that Marsa is fairly calm and even keeled for her X chromosome here, right? And now you're going to take her chromosomes and take them through meiosis, and you're going to get something like this. Okay. Now you can fill in these other ones. I'm not going to fill them in for you, and hopefully you've done your own codings here as well to figure out to make different types of gametes. And then what I'm going to need you to do is take a minute, and I need you to cross the gametes from Marsa that you have made with the gametes of Mars, and I need you to figure out what are their offspring going to be like. So, for example, if we took, one second, let's pop back a little bit. So if we took this first gamete from Mars, right, which is AB, capital A, capital B, that's the chromosome there, and then we cross it with this gamete of Marsa, we get an individual. Whoa, what just happened? We get an individual that is going to love sports, right? Even though neither one of the parents really loves sports that much, that offspring is going to love sports, right? So you should be able to cross the gametes and then figure out what their children really love to do in terms of art, sports, etc. Okay, so you should seriously take some minutes on this, man. Seriously. Trust me on this one. Do it. Okay, I'm going to move us forward. So hopefully from that, you can see that offspring are not identical from parents. And we can start to ask why, right? Why are parents not identical to their offspring? And you get variation in 4 different ways. You get it through mutation, random alignment, crossing over, and fertilization. And this is what we're going to go through. But you also have to remember, this kind of sets us up to where we're headed. It's really important as we're going through this that you appreciate how much variation there really is and how much variation can be generated just from these simple processes that we've talked about of meiosis, gamete formation, and the way gametes come together. Okay, so Mendel had a couple of laws. Mendel's first law was called the law of segregation, and it's a pretty simple law. Basically what it means is that you have members of homologous pairs, right? Members of homologous chromosomes. They're going to separate during the formation of the gametes, and they're distributed to different gametes so that every gamete receives one member of the pair. And all we're saying is you've got all these gametes, right? They're going to come, and during meiosis, they're going to separate in that each gamete is going to get one of these copies, okay? And you can see that pretty well, I hope. Okay, so that's the law of segregation. The next law is the law of independent assortment. An independent assortment means, so it's this process of random segregation and assortment of chromosomes during anaphase 1 of meiosis, resulting in the production of genetically unique gametes. So what we're saying here is that these all assort independently, right? That this could actually be, that these gametes can form this way and this way, or they could align this way and they could assort that way, right? That that just the way that they form and are separated, you get different gametes. Okay, so here I just want to do a little bit of math to illustrate the amount of variation that can come from these really simple processes we've talked about. Okay, so each homologous chromosome is independently assorted, and this is done due to random alignment. Okay, so you have 23 pairs of chromosomes in humans. So that would be 2 to the 23rd, or 8.3 million, almost 8.4 million possible gamete combinations that can happen there. And then when you go, and that's just the formation of gametes in either in either the male or the female. And then at fertilization, you literally have 2 to the 23rd times 2 to the 23rd, or 8.4 million times 8.4 million, and that gives you over 70 trillion possible combinations of gametes when two individuals come together and breed. And here's the exact number for those of you who are sticklers and would like to know. But you have to understand, this is only independent assortment that's at work here. We're not including things like, we're not even talking crossing over yet, right? Imagine this 2 to the 23rd, if you're going to include this 2 to the 23rd here, if you're going to include crossing over, becomes astronomically higher. The possibilities, the number of possibilities for crossing over. And it doesn't include mutation either. So there is essentially limitless variation in the way that gametes form and can come together. So here's this video I mentioned. I think we're going to watch it. It's a really nice review. It'll take you all the way from meiosis through gamete formation. And I think if you follow it pretty carefully, it's pretty informative. Just kidding. Let's take it to the back. All right. So I realized that was a little bit simple, but I hope that helped at least some of you. All right, so let's go ahead and move on a little bit. So the last thing we want to talk about is something called linkage and crossing over. So linkage is when you have two genes on the same chromosome that they don't independently assort. They segregate or move together. And then we ask ourselves, so how would this happen? Why would this happen? And it happens in crossing over. So crossing over can cause genes to separate and move, but linked genes will almost always segregate or move together. And here's how it happens. So if you look at this, right, just look right here. Which set of genes would you guess would segregate or move together during crossing over? Now, if you're looking down here, you're going to get the answer pretty quickly. But I think it's obvious, so I haven't broken this up or anything. But if you're looking at it, you would probably guess that A and B are the most likely genes to be linked and segregate or move together. And that's because they're so close that crossing over rarely splits them apart. Now it could, but it's pretty darn rare that they would split apart. So even with crossing over, these two genes are going to go together. Okay, so not all traits are what we would call Mendelian traits. So not all traits are dominant and recessive. That dominance and recessiveness will display a trait. Some traits are determined by two or even more genes, and these will cause continuous distributions in a trait when they're caused by two or more genes. So some examples in humans that are pretty common are skin color and height. So I will just say this before I move on. I hope that me focusing on skin color makes no one uncomfortable or anything. This is just a fantastic way to look at the genetics of humans and how this works. I should also say that anyone who feels like this gives them any sort of insight into any sort of racist thoughts, totally wrong. That has nothing to do with any human ability in any way. So let's go ahead and continue on here. When we're looking at skin color, actually we'll delve into skin color a little bit more. So what we should take home from this particular graph right here is that when there are two or more traits involved, two or more genes involved in a trait, that trait will have a distribution of shapes or colors or whatever. So looking a little bit more closely at this, if we're looking at individual A and this individual crossing with this individual, you can see right off the bat that they're going to have a distribution of colors among their offspring or children. And individuals that have that end up with all the melanin genes are going to be very dark, and individuals that end up with none are going to be very white. And then you kind of have this distribution of those in the middle. Now, I want you to look at this graph really quickly and explain to yourself, why would there be so many that are going to be in this middle range? Why would there be so many here? And hopefully you're able to answer that. It should be in your book as well. Okay, so here's kind of, some of you probably got really nervous, but here's kind of the answer to that question. When you do, so we did a big Punnett square already, right? Remember, sorry, we didn't do a big one yet, but we did a smaller Punnett square. And then this is just a large one. And what you're looking at is, if you split what's going on here, right, that you've got individuals that can have this as their phenotype, you can have gametes that have this, gametes with this, and this, right? And so if you do a massive Punnett square, you can see right off the offset that about a quarter of the individuals would be fairly light-skinned, a quarter of the individuals would be fairly dark-skinned, and then half of the individuals are going to be intermediate in skin color, which is pretty dang cool. And this can even be shown. So these are twins of biracial parents, and one of the twins came out very light, and one of the twins came out very dark, and they are absolutely adorable. I should also mention that I have a couple of brothers who married Tahitian women, and it's always a big day for me when they have a kid that I get to go in and look at how the genetics came together. I absolutely love it. Super cool. All right, so epigenetics. I don't know if any of you know what this is. I'm just going to invite you to hit the pause button and look it up. What is epigenetics? Okay, so if you looked it up just like I did, first hit that you get is, it's the study of heritable changes in gene expression that do not involve changes to the underlying DNA sequence. And essentially what epigenetics is, it is the relationship of your genetics to your environment. And you'll see here, there's this thing, the new science of epigenetics reveals how the choices you make can change your genes and those of your children. So here's kind of this idea, that you have this underlying genotype that will reveal a phenotype, but your environment can influence that phenotype. Okay, so depending on the food, environmental exposures, drugs, tobacco, whether you exercise or not, the amount of money, the lifestyle that you have, medications, relationships, et cetera, can all influence your phenotype from your genotype. And here's a cool example of this. So if you're looking, so here's a genotype of two plants. These two plants have an identical genotype, but they may grow up in different environments. So there could be environmental influences such as fertilization, sunlight, water, all sorts of things that would make these two plants grow differently in size, even though they have the exact same underlying genetics. And it's not just plants that this happens in, it's also humans. So here are some identical twins, and you have one twin that's literally 2 heads taller than the other, and then in here you got this healthy little fella and his little brother, and his, I don't know, I guess maybe not his little brother, but his twin brother here, perhaps maybe a pickier eater, who knows what the reasons are for this, but it's probably environmental factors. And epigenetics become increasingly complex. We're starting to get a really cool view of epigenetics right now and how this may influence generations down the road. And one of them is, if a mother smokes while pregnant, she not only she essentially affects the genetics of, or the epigenetics of herself, because she's smoking and she's inflicting environmental toxins on herself. But it turns out that because females, if you remember to some of our lectures, females make their reproductive gametes while they're in the womb. So her smoking is going to influence her daughter's gametes, which then in turn would influence her daughter's child's genetics. So there's 3 generations that are affected by a mother choosing to smoke while pregnant, or accidentally smoking while pregnant, or being around it. So in summary, genes are passed from parent to offspring. Some traits are controlled by a single gene. Each gene has 2 or more alleles. And you remember 2 or more, right? Because for blood type, right, we had multiple alleles. And some traits are controlled by many genes. And each of these genes have 2 or more alleles. And then the environment can influence the way genes are expressed. And that is it. A little bit of a shorter one today. Normally we spend a lot of time in the classroom focusing on Mars and Marsa and a few other things, but I think we were able to get through it pretty quickly without working together. I would highly encourage you to spend some time with Mars and Marsa. And I hope you guys have a good day and that everything's going well for you. And we will catch you soon.